Okay, we'd like to start. Okay, everyone. Thank you for coming. So, for our penultimate seminar of the year, we're very happy to have with us Yaara Erez. Yaara received her PhD in system neuroscience from Bar-Ilan University. She then moved to do a postdoc, and she was a research fellow at Cambridge University. Today she's a senior lecturer at the Faculty of Engineering at Bar-Ilan and a member of the Gonda Institute. In her research, she combines fMRI, ECOG, single-cell recordings, behaviour and computation methods, both to do basic science and to do clinical studies. And today we'll hear about individual-oriented cross-modality functional mapping of the human brain during executive control using fMRI and ECOG. Thank you for coming.
Thank you, Jonathan, for the introduction and for the invitation, and I'm very happy to be here today. Jonathan already mentioned the title of my talk, and before I start, just a few words about myself. I'm now based at the Faculty of Engineering at Bar-Ilan, and I opened my lab there just a few months ago at the beginning of the year, after spending around 9 years at the University of Cambridge. So I'm learning Israeli again now. And all of the research and work that I'm going to show you today is work that was done in Cambridge and together in collaboration with Cambridge University hospitals. So just a few words about the research in my lab. What we are really interested in is how to use neuroimaging at the individual level, or what is sometimes called precision neuroimaging, and I'm going to talk about it today. In terms of the cognitive processes that we focus on, we are interested in attention, how we extract relevant information from the environment and filter out irrelevant information, and more generally other processes that are related to executive control and the brain system that support them. We are using a variety of computational tools and imaging modalities, just like what Jonathan mentioned, fMRI, ECOG, simulation, now getting into virtual reality to try and understand these processes in a way that is more similar to how we experience it in our daily life. And we have a central branch in the lab where we develop clinical applications from the knowledge and tools that we have from basic science research. And I'll start with the take-home message from the talk, three main points. The first one, I hope that I will be able to convince you that it's both important and possible to look at individual level neuroimaging data. The second point is about our ability to integrate data across imaging modalities. Most of the research focuses on just one modality at a time, but we can gain many more insights if we have better tools to combine across modalities. And the third point is about the relationship between basic science or neuroscience and clinical applications. We usually think of it as a bidirectional arrow from basic science, then translating it into clinical applications. But actually, when we do clinical research, we have sometimes amazing data that we can learn from about neural mechanisms. So this is in fact a bidirectional relationship. So we have many different brain networks or regions that work together, and they support different functions. This is data from Gordon and colleagues, just to demonstrate some functional networks that we have, very colorful. I highlighted a few that I'm sure all of you know. And this is an average across a group of participants, and we can see those different networks. And what we want is to be able to understand what they actually do. And we have many challenges in the field, but for the purpose of this talk, I want to highlight a couple. The first one is that most of the research is done across a group, as an average across the group at some level. So we always end up averaging across the group. And in neuroimaging, in many cases, it depends on the system that you look at. We look at the same regions in all individuals. But when we look at individuals, and this is data from the same paper as the previous slide, but this time in individuals, we can see that actually the functional networks are much more detailed. It's useful to average across participants because then we are left with a signal and get rid of the noise. And the challenge here is to be able to look at it in individuals, but still separate the signal from the noise. And that's one of the challenges that we deal with in the lab. And the second challenge that I already mentioned in one of the points in the take-home message is about the integration of data across imaging modalities. We should all strive to do that. Every method measures something slightly different, but if we can combine data across methods, we can benefit from the advantage of each method. And I'm going to show you in the talk how we address each of these challenges in our work. And I'm going to show you while focusing on executive control or cognitive control or executive functions. You can choose whichever term that you like. In this picture of kitchen service, we see the people making a meal. This is something that we all do, right? But in fact, from a cognitive perspective, this is a very complicated process that involves many, many aspects. We need to attend to the relevant information, filter out irrelevant one. We have to plan ahead, multiple steps, tasks. We have to solve problems when they arrive. We have to switch between many different tasks. And this demonstrates all of the range of what we term as the family of executive control functions. And it's not, of course, just about preparing meal. This is something that we all do most of the time effortlessly in our daily life when we plan a holiday, host a party, pack our bag in the morning, and so on. At the neural level, we know in the human brain that a network of regions that work together in the frontal and parietal cortex support these functions. So we have, I hope you can see my photo, a strip along the middle frontal gyrus, an area just anteriorly to the frontal eye field, the anterior insula, and the IPS. This is a visual component that is not considered as part of the network, but it's here because of visual tasks were used here. And on the medial surface, the pre-SMA. This is a bilateral network. It's the main general, so we can see the recruitment of this network across many cognitive domains, like numerical, verbal, non-verbal, and so on. And we can easily identify it when we use cognitive demand manipulation. So whenever we try hard, this network is recruited. It has been associated with many aspects of cognitive control that I mentioned earlier, attention, planning, and so on.
Can I say something? So I also see the cerebellum more.
This, sorry?
The cerebellum.
Yes, a little bit. Okay. You don't have the cerebellum here, but actually in the paper, so this domain general network is being updated, so it's not anatomically well defined, and there are efforts to do that. This is just one other example to try and get a better and more anatomically accurate of what regions or which are the regions that are actually included in this network. There are also some subcortical regions that we don't see here.
So cerebellum is included, part of the cerebellum?
It depends who you're asking.
I'm asking you.
Yes. No, I mean in terms of the literature. Usually we don't include it in this network, although we do see blob in the cerebellum here. But I would say that it's not necessarily because it's not part of the network, it's because that we usually ignore it, and it's worth a deeper investigation. So with this in mind, I'm going to talk about two main studies today. The first one is our attempt to localize cognitive control regions in individuals with fMRI. These are healthy individuals. And the second part is our efforts to map executive control regions, this time with patients that undergo surgery for the removal of a brain tumor and using electrocorticography or ECOG, recording directly from the surface of the brain. And I'll start with fMRI. So what we normally do when we look at this network of executive control, we look at the same regions for all participants. But what my former students, Sneerhan and Florger, were interested in is to actually see whether we can localize these regions in individuals. And for that, we systematically assessed this between and within individuals using 3 different tasks, all tasks that have been previously used in the literature of cognitive control, spatial working memory, verbal working memory, and a Stroop task. And because we can identify this network with increased demand, all of these tasks had 2 main conditions, an easy condition and a harder condition, which is basically exactly the same task, just more challenging. And when we look at the change in activity between these 2 conditions, we can see these are group averages. We can see the recruitment of this typical pattern of the executive control network. So this is a good sanity check. But when we look at individuals, unsurprisingly perhaps, we can see huge differences between people. So these are examples from 6 different participants. I highlighted a couple of places where there are differences, but there are more. And I hope you can still see this typical frontal and parietal pattern of activity, but you can also see the differences, which we usually ignore. So we decided to look at them. So to quantify these differences, we started by looking at how this looks like across individuals. This is not the standard fMRI activity map. This is a probability map. So the color of each voxel indicates the number or proportion out of 15 of participants that had a significant change in activity with increased demand in this voxel. So the yellower parts are where most of the participants had such a change in activity, and the reddish or darker parts are where a smaller proportion of participants had such a change. And we can see 2 main things. First, we can see how widespread this map is, reflecting the variability across participants. But we can also see the yellower hotspots showing this more typical group average that we normally see in typical studies.
How stable are the maps within a subject?
I'm glad you asked. So to further quantify this, we wanted to assess how reliable the patterns of activity is within and between participants. So the darker bars are between participants. Each participant repeated each of the tasks twice, and we correlated activity across voxels. These are the reliabilities for each of the 3 tasks, and you can see how high they are. That's quite impressive for fMRI. So whenever we repeat the same task twice, pretty much we get the same pattern of activity. But that's not the case across participants. These would be the lighter bars. Now we correlated across voxels data from 2 different participants and all possible combinations. And you can see this drop in correlation reflecting the variability across participants. What we wanted to ask next is whether this at all reflects something more general about executive control. So if participants repeated the same task twice and we got a similar pattern of activity, that's nice, but maybe that's just very specific for this task. So what we did is we used the data, the patterns of activity from each of those tasks as localizers together with another independent criterion task that each of the participants did. And we looked at activity within those individually defined regions. For that, we used a task switching task, another core aspect of executive control. And as a start, we used what we normally use in the field, the group template, to define these regions. What we expect to see in this task is an increase in activity above 0 for switch compared to staged trials. And we were able to replicate that in our data. This was published before by another group, so that's nice. And next, we wanted to look at what happens within the individually defined regions. You can see where this is going because the Y scale is larger. So not only we were able to see the same effect, but it was stronger. So sensitivity was larger, and this also demonstrates that those regions that we were able to identify in individuals are more generally related to other aspects of executive control. And we can use the same method in other aspects and other ways of looking into fMRI data, for example, decoding with multivoxel pattern analysis or MVPA, where instead of looking at average activity, we look at pattern of activity across voxels in a given region. And the idea is that if the pattern across voxels is meaningful or in any way or represents something about a specific condition or 2 conditions, then we will be able to look at the pattern and then decode which condition each pattern belongs to. And this is, and if we can do that about the chance, about 50%, this means that there is some information that is represented about the cognitive process that we look at in these areas. So we did that this time with an attention task, and we're able to show that only relevant information is represented, but not irrelevant one, which is what we would expect from a network or regions that are involved in attentional control. And I'm not getting into all the details of this task here, but I'm happy to talk about it later on. We also looked into this attentional control process in more detail with single cell data from the lateral prefrontal cortex of monkeys, and looked at subspaces that are task related and how the temporal profiles look like within those subspaces. I'm not going to talk about it today, but happy to talk about it later. To summarize this part, I showed you that the executive control network is unique in individuals and can be reliably localized. And this is particularly useful when we work with clinical populations, because when we have a patient and we want to provide a good treatment, we want to provide treatment to this individual patient and not to the non-existent hypothetical patient. And that brings me to the second part of the talk, where I'll show you some data that we have from patients during awake surgeries, And we used ECOG to try and identify executive control regions. Yeah.
The repetition of the MRI measurement was in the same day?
Yes.
Okay, so this implementation will be interesting to see. It was like 1 month to see if then it is more give more reliable estimation of the user variability.
Yes, I agree about that. I think there are some works that look at that, but not in the same way. So they don't look at reliability of this particular network. They look at cognitive processes and how reliable they are over time. This project started a few years ago with an email that said, well, just before I deleted, are you interested? And then I said yes, and this is how it all started. And we worked with patients with diffuse low-grade gliomas. There are many different types of brain tumors. Diffuse low-grade gliomas account for about 15% of primary brain tumors, tumors that arise in the brain. You can see an example here in the darker gray block that I hope that you can see in the frontal cortex. This is one of our patients. The average age at diagnosis is rather young, and the average survival is rather long. So these are non-aggressive tumors. This is good, but they also spread into the healthy tissue around them. So there is no clear boundary between the tumor and the healthy tissue. This is why the non-aggressiveness of the tumor means that the average survival is long, and this is why it's important to provide the best quality of life for patients. They can now live with these tumors for more than 20 years, and basically we want people to have normal life. The first line of treatment is usually surgical intervention for the removal of the tumor. But because there is no clear boundary between the tumor and the healthy tissue, we need to optimize a very fine balance between removing as much as possible of the tumor on one hand and preserving function, any function, motor function, walking, writing, any aspect of language, and any other cognitive aspect. And the way this is now done routinely mostly for motor function and some aspects of language is using an awake surgery procedure with stimulation. So patients are being awakened after the skull has been opened, and then they are asked to do simple tasks like counting or looking at pictures and saying what they see. And while they do that, the neurosurgeons apply brief pulses of stimulation in different parts of the exposed tissue. The idea is that if stimulation leads to impairment in this function, for example, the patient stops talking, then the stimulated area may be critical for this function, in this case, speech production, and then the neurosurgeon might decide not to take it out. But while this is done for motor and some aspects of motor function and some aspects of language, it's much more complicated for executive functions for two main reasons. One is because of the complex nature of these functions, and the second is because of the distributed network of regions that support them. When we started, actually even several years after we started, there were only 3 papers published in the literature with an attempt to map executive control regions during surgery using stimulation. They all used the Stroop task, another aspect of executive control where people need to read a name of a color colored in another ink color, and what they need to say in this case, they see the word blue, but they need to name the ink color, which is green, and suppress reading the word. This is actually quite tricky, and I know that many of you are familiar with this task. And our approach was different. What we suggested is that we can use ECOG to identify the regions that are involved or associated with executive control. Stimulation is very useful, but one of the problems with it is that it's very time-consuming, so neurosurgeons need to quite blindly move from one area to another, repeat it several times. It has the risk of seizures, so we really want to provide tools to make this process more efficient and safer. And we suggested ECOG for that. Our aim was to identify a neural signature of executive control regions, and of course this is a very coarse neural signature. Our research has been featured by the BBC several years ago, and I'm going to show you now a short part from this video. You can watch the full 9-minute one on the lab website. This is Ben. He was in his late 20s when he was diagnosed with a low-grade glioma, and he has very kindly agreed not only to take part in our research, but also to be filmed for this feature by the BBC. And I want to highlight a couple of things in this short clip. In the first part, you will see Ben talking, and then the neurosurgeon will apply stimulation. You will see what happens. And in the second part, you will see Ben participating in our research, and you will also see me there. So let's see.
A few months later, surgeon Thomas Santarius is removing it, while Ben is still awake.
Okay, you take a break a little bit.
We were even allowed to talk to him.
I know these are very strange circumstances. You've been doing amazingly well, and you've still kept your sense of humour.
Yeah. Try to. You have to laugh, eh?
Using an electric current, Thomas could switch sections of the brain off. Listen to this.
Can you count for me again?
1, 2, 3, 4.
The fact that Ben stops counting means it's a part affecting speech, so Thomas won't remove it.
We'll start now.
But it's not the awake part that makes this operation special.
A1, B2, C3.
Yaara Erez isn't a doctor, she's a neuroscientist.
F6.
This test is hard enough when you're not in the middle of surgery. You try doing it.
8, 5, 9.
By forcing Ben's brain to multitask, scientists are creating a much more detailed map of its functions for a new device that will eventually help surgeons decide what to take out and what to leave behind.
So I hope you could get a flavor of how this looks like in the operating room. And now I'm going to tell you a bit in more detail what we actually do there. This study was done together with my former student Muatas, and this is how the operating room setup looks like. We have the patient lying down here. Electrode strips are placed directly on the surface of the brain. This is after the skull has been opened. They are connected to the acquisition system where we record the data. We use the neuronavigation system with this probe to get the locations of where the electrodes are placed. And we record this data while the patient is doing some simple cognitive tasks. The ultimate aim is to provide neurosurgeons with feedback in real time, but as a start to establish this approach, we record the data and analyze it offline, and all the results that I'm going to show you today is from such offline analysis. And what is special about this signal is that we have both high temporal and high spatial resolution that we can't achieve with non-invasive methods. So what kind of tasks can the patients do? There are actually many constraints for a task that patients will be able to do in the operating room. I will not get into all of them, but what we ended up deciding about was to use a similar approach to what we can do with fMRI, with this idea of cognitive demand manipulation, an easy and a harder task to recruit the executive control region. So instead of tapping on a specific or particular aspect of executive control, what we're trying to do is to recruit those regions. So the easy task was counting from 1 to 20, and the harder task was counting, but this time switching between letters and numbers. So 1A, 2B, 3C, and this becomes very hard very quickly. It also has a task switching aspect for executive control in it, and similar to the trail making test for those who are familiar with it. And what we're looking for is a measure in the ECOG signal that will serve as this neural signature. What kind of measure? Again, we didn't know at the start, but based on previous literature, what we decided to look at was power in the high gamma range between 70 and 250 hertz. Why we thought this would be useful? So this power in this frequency range has been previously associated to cognitive processing in other domains like the motor domain, visual domain, auditory domain. It has also been associated or linked to the fMRI signal, volt signal, and spiking activity, and has been used in the brain-computer interface. And based on all these previous studies, what we hypothesized or hoped to find was an increase in high gamma power with increased cognitive demand. So when we look at the change in activity between the hard and the easy counting task. So as a start, example from 3 patients. Each dot here is 1 electrode, and they are color-coded based on their on the change in high gamma power with increased demand. So red means increase in high gamma power, blue means decrease. They are all placed on the frontal cortex, and we see that at least in some of the electrodes, we can see this increase in high gamma that we expected to see. And the next step was to do the same thing, but with a larger sample. And this is what we did. We recruited 22 patients, had complete data sets from 13 of those patients, and a total of 79 electrodes covering the frontal cortex, which is an area that is not very common for ECOG data from epilepsy patients. All the electrodes here are displayed as white dots on the left hemisphere, but we had some electrodes on the right hemisphere as well. After re-referencing, we are left with 59 electrodes. And just for visualization, what I'm showing you here, red again is increase, blue is decrease, and you can see the overall activity across our sample. There are some electrodes that I'm not sure if you can see well on the motor cortex, and what we can see is this red blob in the frontal cortex, but not more posteriorly on the motor cortex, and just before that. We further looked at the activity in each electrode individually with a statistical test that we developed because we need to preserve temporal correlations here. And you can see that now only the electrodes that showed such significant change are colored, the rest are in white. And you can see that the vast majority of electrodes with significant change showed this expected increase in activity. Together with the increase in high gamma power, what we in many cases see is a decrease in another frequency band, in the beta band. So we also looked at this frequency band, and we saw these expected decreases, but this time instead of the focal increases in high gamma, we see decreases in the beta that are much more widespread. So high gamma increases are focal, beta power decreases are widespread. And the next step was to more formally link it to executive control regions. What we did for each electrode, we determined the network that it belongs to. This is based on resting state fMRI data from the HCP data set. You can see in the different colors which electrode was linked to which network. In orange, it's the frontoparietal network that we look at, but you can also see the DNN in red, the language, and so on. And then we looked at several measures for spectral changes within and outside the frontoparietal network. So to start, we looked at the proportion of electrodes with significant changes. In this figure above the zero line, here, these are the proportion of electrodes with significant power increases. Below is power decreases. And in this yellow bar, you can see that a large proportion of electrodes within the network, within the frontoparietal network, showed such significant increases compared to a much smaller proportion outside this network with similar increases. So increases are much more likely within compared to outside the network. And for the beta range, decreases are pretty much equally likely both within and outside the network. And we looked at several other measures. I'm only going to show one. The picture is very similar. This is just the overall power change across all electrodes, not just the significant ones. And you can see increases within the network, but not outside in the high gamma range, and decreases both within and outside. To get a slightly more detailed view into how these changes look like for the different frontal networks, we parsed it into those networks and the electro data that we had in each network. And we can see that only the frontoparietal network shows these increases. So these increases are specific for this network, and we don't see them in other networks. Yes.
Just making sure areas here were predefined based on the HCP, not based on some subject localizer or something like that.
Exactly. So based on the HCP data. What I want to highlight here is that two things. First, we have some converging evidence from fMRI data in an independent data set in our ECOG data. And the second point is about the importance of anatomical, taking into account anatomical locations when we use ECOG data. So in many studies, we use very general labels like the dorsolateral prefrontal cortex, but actually, if we look more closely at the different networks, we may see different profiles of activity. Overall, our results suggest that high gamma power is a good candidate measure to identify executive control regions. What I showed you so far is data from electrodes that were placed on healthy tissue further away from the tumor. But what Ayan, another student from Cambridge, what Ayan was interested in, what happens on the tumor itself. And we had just a few electrodes that were placed on the tumor. It's actually on the peritumoral area. It's not in the core of the tumor because it's always on the surface. And he looked at that. There is some emerging evidence from the last 2-3 years, mostly at the circuit and molecular level, for the integration of the tumor with the healthy tissue around it. And we had the opportunity here to look at it in terms of functionality in patients during surgeries. I'm showing, this is still work in progress. We are now writing up the manuscript. I'm showing you only a couple of examples from 2 patients. These are 2 electrodes that show this significant increase in high gamma with increased demand. They are both on the tumor. You can see this slightly blurred area over here. This is, I hope you can see it. This is the tumor area. So we were able to identify some functionality there. But more interestingly, We also had resting state fMRI data for all of the participants. So what Ian did, he looked at this resting state data with the electrode as a seed region. So what he wanted to see is whether there is some preservation of functionality not only locally but also at the network level, at the whole brain, whole brain and network level. And he saw that some functional connectivity is preserved also at the whole brain level with executive and attentional networks. So to summarize, I showed you that we can reliably identify control regions in individuals with fMRI, that we can use high gamma power as a coarse UL signature to identify these regions with ECOG during surgeries, and that there is utility for using cross-modality integration, both when we look at ECOG data within predefined networks, and also when we combine ECOG data with resting state data, fMRI data within the same patient. And going back to the 3 points of the take-home message, I hope that I was able to convince you that it's both important and possible to look at data at an individual level, that there is a benefit in combining data across neuroimaging techniques, and that this bidirectional link between basic science and clinical applications is also beneficial. As we are a new lab, we are recruiting. I was told that most of the students here are graduate students anyway, but if you have friends, please tell them that we are recruiting at all levels, and I will be happy to hear from you. On the top right is the lab website and my email. Thank you very much for your attention, all of the collaborators in Cambridge, and particularly the patients who agreed to take part, and I'm happy to take more questions.
Yeah, 2 questions. The first of the 2 talked about initially about several tasks that are related to executive control, and you showed us that within a subject and within a task you get repeatability, which was relatively high. R was 0.7 or something like that. I was wondering if it was also the same across tasks within a subject. So that's question number 1. And the second one, which is actually more important, is, okay, let's say that you identify the electrodes which are involved in a specific task, okay? And it doesn't really matter what task you use it, because the answer to my first question is yes. Still, the question is, would you ablate this position? Because you said there's a network, okay? So maybe ablation of a specific area doesn't have much of an effect on the patient's ability to perform later. So, which is really the question you're interested in. So.
Okay.
Okay.
Thank you. Excellent question. So to answer the first one, yes, we do see a similarity. It's slightly lower. I can't remember the number, but we did look at it in the paper. So there is some correspondence, and it's above what we see across participants. So it doesn't go as low as that. But there is a slight drop, and it also depends which tasks you are comparing. I don't know if you remember, but even in the figure that I showed for the working memory tasks, replicability was higher. It was slightly lower for the stroke task. So when we look at replicability across the 2 working memory tasks, we see higher replicability compared to working memory task in the stroke. So that's to answer the first question. And there is still work to do there, right? So it's not perfect replicability, and there are many other aspects that we can look at, including long-term replicability. To answer your second question, of course, you are absolutely right, and that's exactly the question that we are dealing with. So when we identify regions, the aim at the moment is not to tell neurosurgeons, well, don't take it out, but rather to point them to areas that they might want to use stimulation on. So instead of just looking at all the exposed tissue, they can focus on just specific areas. And we now look at whether this is indeed the case. So do we at all see response to stimulation within those areas more than outside what we identified with high gamma power? And we don't have the answer yet. But also, I want to emphasize here that also stimulation, it is used as the gold standard. And when I talk to neurosurgeons, there is not so, and I understand that because that's the that provides causality, right? But even stimulation, you have different parameters. Maybe you are not using, so it may be different for cognitive control compared to motor. There are many questions there. So even stimulation, it is the best thing that we have at the moment, but it's not perfect. And it could be that. It's a very long answer, but nearly there, that you will not see an effect with stimulation, and you will decide to take this part out, but only later on you will see some deficits. So I don't have the answer yet. We didn't do a randomized control trial, and at this stage I wouldn't dare to do it yet.
Yeah, but so you probably want to do a longitudinal study in which you follow up these patients to see, okay, which...
Exactly. So we have some longitudinal cognitive data for these patients. We can talk about it later. There are a whole host of issues there as well. But here we didn't, the clinical decision making was not based on our data.
Are the typical parameters for the stimulations?
Sorry?
Did you say there are the typical parameters for the stimulation?
So it's mainly the intensity that they change.
Not the frequency?
Not the frequency. They use, it depends on the neurosurgeon, but there are a few typical ways of using this, using stimulation during surgery. What they do is first they establish the threshold for each participant, and they do it while testing in the motor system.
The thing is that we know that the intensity, the threshold intensity relates to the frequency. So it's not two independent parameters. That's why I asked.
Yes. So there are a few frequencies. It's a very short train of between 4 and 6
the
pulses.
higher the frequency go.
Yes. So it's always between 4 and 6 and pulses, if I remember correctly. It's very short. It's a very short train, and they don't. I think, I'm not sure about these details, about these 4 to 6, but yes, during surgery they don't change the frequency, just the intensity. They do work with either monopolar or bipolar, which is also slightly different in terms of what it does, but yeah.
I would inquire with them about it. No?
I can try.
We just talked about the frequency of the simulations before the seminar. Okay.
So how much do you think those differences between individuals can actually be attributed to differences in performance? So what happens, for instance, if you quantify, or use it as a covariate, etc.?
Yes. So there are some studies. We didn't look at that with performance. We didn't have enough participants for that. But there are some studies that look at that. You do need quite a large sample for it. I only know about one study with a large enough sample. And I must say that what they found there is opposite to what I would hypothesize. So larger, and they only looked at the average total change in activity, not the reliability of the network. And they found that with the higher or the larger the change in activity was with increased demand, it was positively correlated with performance such as accuracy, with measures as IQ, and negatively correlated with reaction time. I would actually hypothesize the other way around. For example, if we think about it in terms of working memory, then perhaps if someone has a larger working memory capability, then maybe there will be smaller increase in activity. So there is that one study with a large enough sample, but I don't think it's a solved issue.
I guess the kind of provocative question would be, okay, so let's assume that you know a person's IQ performance levels. Would you then be able to pinpoint the specific area and the extent of that area that's involved in that particular function?
I don't think it's the spatial distribution of the network. In my view, I don't think that's related to performance. If at all, it would be the activity within those regions, or what kind of information, or to what extent information is represented in those regions, but not. Why would you think that it would be related to the spatial arrangement of the?
When you're trying to account for those individual differences, you have to account, first of all, for anatomical variations across, people's brains. But secondly, you want to make sure that it's not related to just differences in performance, because obviously those will contribute to the extent of activation. So you want to make sure that you're reporting that.
Right. So you mean that how not necessarily if it's the same amount of activation but in slightly different crowd, but how much of activation, how widespread it is, or how large it is within those regions. So you're right. There are several things that we can look at there. I don't have an answer.
I mean, could you just use RT as a predictor and then see whether that would overlap with those particular areas?
Yeah, we can do that. In this sample, we have the data, but it's not a large enough sample, I think, for correlative measures.
I want to ask about combining MRI and EEG or ECOG. You aim also to have better spatial and temporal understanding of the brain that will assist you also to find causality in between the network?
What do you mean by causality?
Because the MRI give you more correlations, and the EEG cannot exactly give you the locations. Now they try, but still it's not accurate. So when you have both, in theory, you can map the network and you can really see the temporal relationship in the network, and then you can understand more the causality.
Yes.
This is one of the aim of your integrating the information?
Yes, so it depends what you integrate with what. So yes, if you integrate MRI and EEG, and there are some simultaneous, not my work, but there are some studies where they simultaneously recorded MRI and EEG, and then you get the spatial precision from MRI and the temporal precision from EEG, and you can combine them. You can do that with MRI and EEG, not at the same time, but you can. There are now methods to they call it fusion MEG fMRI. So there are different ways. But yes, for example, what I showed with the ECOG, so we can get the very local activity, but then with fMRI we can now see what happens in more distant regions.
I'm trying to figure out what is the message, or what is the conclusion we should make from the inter-individual differences, in the sense that it couldn't be different, right? So our brains are folded a little bit differently, so even the gross anatomy varies a little bit based on our genetics, and then the functional mapping into the gross anatomy also varies. So not the same functional unit can be at the crest of the gyrus or at the bottom of the sulcus and so on. So just by that, you would expect that within subject you'll have a better correlation than cross subject. So I can see the merit of defining methodologically, so saying like an individual localizer is better than a group localizer. But is there also something that we should make of it in the more for understanding inter-individual differences, or into understanding the mapping of function into structure?
Yeah, so I think, so all of the points that you said are of course true. That's how I usually start that talk. And the fact that we see individual differences is not surprising at all, right? I would be surprised if it would be otherwise. But my point, at least in this talk, was that usually, at least in the cognitive control domain, this is not normally done. And we have the reason why we actually started with this study was my frustration with the very low decoding results that we have in the frontoparietal network. If anyone here is doing this, then you know what I'm talking about. And you also saw in my data, we reached the 52%. It's significantly above chance, but it's very low. And that's what we normally get. So it's not unusual. And this led me to ask, well, are we at all looking in the right place? So I think that the methodological benefits of looking in the right place will also lead us to perhaps see things that we can't otherwise see. So that was the driving force for this study. I don't know if I answered your question, but that's where this is going.
So do you find that in decoding?
No. We did see an increase in the univariate data. We didn't, in this very systematic investigation, we didn't see an increase in the coding, but we did see it not in a controlled way in other studies. So I have several studies where we use the decoding, and whenever we look at individually defined regions, we see this slight increase in decodability, but not when we did it like in a very systematic, controlled way. And perhaps to continue your questions, the question that you didn't ask, we then asked ourselves, oh, okay, so what does this mean that we don't see, that we do see increase in univariate activity, but not in decodability in terms of the underlying architecture and how these regions work and so on. I don't have a definite answer. It's an open question for discussion about which I'm not sure that we can totally answer with fMRI data. But what does it tell us about how these regions are organized and how they work and how information is represented?
Probably the spatial resolution of fMRIs.
Sorry?
The spatial resolution of fMRI is probably not suitable for that.
Yeah. That's one reason. I don't think it's the only one, but yes.
Okay, thank you very much.